To begin this discussion, I'd like to remind you of those sets of experiments that were done by Speeman and Mangold. So remember, they were working with salamander embryos, and they were doing transplantations and ablations. An ablation is basically just where you kill cells with a hot needle or cut off a few cells so that they're no longer there. And they came up with this idea of what they call an organizer, a group of cells that somehow signaled to the cells around it what they were going to become, and that these help to form an animal. So if you take an organizer that helps to form the dorsal side of an animal and transplant it onto another animal, so now that it has two of those, it ends up with two signals saying, hey, be dorsal cells, and it recruits different parts of the embryo to become that, and we essentially create a second embryo from that one small group that organizes all of the cells that it comes in contact with. Okay, so that's the summary. Now, at the time, they didn't know what that organizer was. So as we began to go through the modern synthesis, understand DNA, the role of genes, we realized that it had to be genes, or more specifically, gene products, right? So the organizer creates a signal. This was sometimes called the morphogen. So just be familiar with those terms, right? A morphogen is something that induces cells around it, right? Signals to those cells to change their cell fate or to direct them down a certain fate. And so we can look at building an organism as an embryo as a series of organizers acting upon other parts of the animal, maybe even then creating new organizers downstream. Those work, and we subdivide, and we finally get a fully formed organism. Now, that's a very simple overview of an incredibly complex process. And we're going to do some of it, talk about it, and especially in the terms of changes between big organisms. That's our focus for this class. Okay, so the organizer creates these signals in the form of mRNA. That's the most common one. The mRNAs can then diffuse across a growing embryo and send a signal. Proteins obviously are the final action, and most of these mRNAs that are important are either signaling factors or they themselves are transcription factors. So in blue and this kind of fluorescent green color here is a three-dimensional diagram of a Hox gene. Hox genes bind to DNA, and by doing so, they can activate genes or repress genes. So they change gene expression. That changes the fate of those cells in early development. And a single gene in the right time, or the lack of a single gene when it should normally be there, can make a massive change downstream. The earlier in the developmental sequence it is, the bigger the potential change downstream. And again, these changes, like they were doing with the fruit flies, are just random mutations. Most of them, the vast majority of them, are negative. They change morphology so drastically that there's a very small chance of it being a beneficial change. But in the laboratory, we can maybe keep them alive, or at least observe what the potential impact of it was before the embryo dies from that major mutation. Okay? But it's a window into the developmental toolkit that builds these major structures and can make big differences between species 1 and species D. So that's a morphogen, right? A morphogen is any signal that can induce, that can change the fate of the cells around it. And so you need to understand that those morphogens are gene products that are either signaling factors for transcription factors, or they themselves are transcription factors. Okay? And again, a definition of a transcription factor is a protein that binds to DNA and regulates gene expression. Okay? So let's look at a model, and then we'll look at one specific example with a couple of different morphogens that help to form a limb in vertebrates. Okay? So here is our model of how these morphogens work. You can kind of think of them a little bit like a light switch, a dimmer switch, right? So when we think about genes, it's not simply is that gene turned on or is it turned off? It's also about the timing, right? When is the gene turned on or turned off? And then it's also about how much of it. Do we get a little bit? Do we get a lot? And if you have a little bit of a gene, sometimes you can have a very different end impact than if you have a lot of a gene. And sometimes there are ranges of different results depending on the amount of the gene that is there. And so this idea of a dimmer switch, or maybe even better, a gradient, is key and critical to understanding how morphogens work. So for this model, we're going to use one cell or maybe a very small cluster of cells that begins to express an important toolkit gene, and it is a morphogen. And what happens is as more and more of that is made, it begins to diffuse away from the primary source. And initially, we're just going to do this in a single dimension. We're just going to do it in one direction. So let's say we're on the end of a group of cells here, and this one cell starts making this gene product. And now we know, again, it's an mRNA or maybe a protein, but mRNAs. And those mRNAs then begin to diffuse away. And so if we look at the concentration of these mRNAs, they're going to be incredibly high, of course, where they're being made. But then they're going to steadily drop down and drop down and drop down until they become nearly, there's a certain point where they're no longer able to diffuse that far. Now, mRNAs have a fairly short life, so that can determine how far they're able to diffuse before they get broken down and degrade. So that can play a role also. And once those mRNAs get into one of these target cells, they are then translated into the protein product. The protein product then does whatever its job is. So notice that we have a gradual reduction in the level of this morphogen the farther we get away from its source. Okay, fairly straightforward. If you want another analogy, you can kind of think of it like if you have a pizza in a room, the smell of that pizza, those little volatile molecules that make pizza smell so good, are coming off the pizza. It's going to be strongest right next to the pizza. And as long as that pizza is hot and making that smell, you're going to have a strong smell there. And then the farther away you get, you're going to have weaker and weaker smells. Now, of course, depending on the size of the room, eventually you might saturate so all of the room has the same amount of smell. But in cells, there's usually enough room to work with that we get a fairly consistent gradient going away from the source. Now, this could also work in in 2 dimensions, and we'll talk about that, and of course in 3 dimensions also, and we'll look at that in later examples. But for now, we're just doing it along this one dimension. Now, another key concept to how these things work is not only do we set up a gradient, but then there are also threshold values that depend on how much of that transcription factor or signaling factor is present in the cells. So if we drop below a certain point, then it's almost like we have flipped a switch. So the gradient itself is a little bit like, not exactly like most of our dimmer switches on lights in our homes. It's really more of like you put pressure and pressure and pressure, and then it would go, and then a little bit more pressure, and you'd get to another state. So once we reach a certain threshold, boom, we have a different cell fate. So all the cells next to it that get a very high signal have one fate. All the cells that are getting a medium level have another fate. And then the ones that are below the other threshold point really don't change at all. And so by having one single signal with a gradient and different threshold points, we can set up 3 different cell fates based on that one signal, or even more in some cases. And again, it's going to depend on the interaction, how many binding sites there are, how many target genes, and we'll talk in more about some of those specifics later. And so we're going to look at one example of this, and we're going to use one that was discovered, and these organizers, these cells that we're sending out, these morphogens, were given a very specific name, and they've got some specific phenotypes when they are manipulated. But before we do that, we're going to do this final thing where scientists begin to break these problems down, and so they started to think about these morphogenetic fields with both primordia, and then also secondary fields. So we can basically kind of subdivide animals into all their different parts. We maybe even want to think of them as modules, and then look at the genes that are responsible for those specific parts. So for instance, the fruit fly wing could be treated as a single morphogenetic field with a series of genes being made and expressed at different parts, diffusing out, and then having impact depending on their concentration and on the threshold levels, having different impacts and different cell fates. So just be aware of the term primordia, secondary field. These are basically all similar terms for a morphogenetic field. But then we may have tertiary and quaternary and different subdivisions of those original fields. So the biggest primordia is just the animals itself. And we might divide it, as we will show you later on, how we divide it into anterior versus posterior regions, how we divide it in dorsal versus ventral. okay, so that we can then further subdivide and subdivide, and different cell fates can then be locked in for those different regions of those subdivided morphogenetic fields. Okay, and so these different compartments then are subdivisions of the original field. Now, a key idea here is, this is related to human health. The term that we see more frequently when we're talking about human health or other interventions is stem cells. So realize that all of the cells in an early embryo are stem cells, or maybe more specifically, embryonic stem cells. They basically are blank slate, and they are waiting for signals that come from the neighboring cells, and if those signals all work correctly, then we get the original morphogenetic field established, subdivisions, further subdivisions, and continuing cell growth and division and patterning until finally we get the fully formed organism. So a technical term for stem cell is a pluripotent cell, a cell that has infinite potential to become any type of cell in the body, but we have to get the right set of signals, the right complex amounts and timing to result in that. So in theory, we could take a single cell that was an embryonic stem cell, let it divide, let it grow, and if given the right signals, we could grow a human heart. So that's an incredibly powerful idea, but it's a very, very complex process that relies on all of the signals around it. So trying to take that process and do it in vitro, right, out of the developing organism is incredibly complicated, and we don't have the level of sophistication or understanding of the process to do a complete heart. Hearts are, connective tissue and muscle tissue and nerve tissue, and all of these things integrated together in a very complex manner. But it happens naturally, and sometimes it goes wrong, and we end up with defects, or we end up with unviable embryos. But it happens naturally in the developing embryo. Now, the early experiments that we're going to look at show that there were two types of organizers in limbs, and we're talking specifically, in fact, let's modify this. We're going to talk specifically about vertebrate limbs. Although, again, we might expect to see some amount of similarity if we were looking at even more distantly related things because of this surprising conservation of these toolkit genes. But this one is specifically for vertebrate limbs, okay? The first one that was discovered was called the apical ectodermal ridge. So you need to know that name and know what it does, okay? The apical ectodermal ridge, apical, just two ways to pronounce the same one, creates a signal, a morphogen. Don't worry about the name for this. Maybe I'll put it up here, but I'm not going to hold you to it. I don't want you to memorize this, but I do want you to look at the results. So the signal is, or let's just call it the morphogen, is a gene called FGF, which is something growth factor -8. Many mammalian ones, because they were discovered later in large groups, don't have really inventive or helpful names. But this growth factor 8 is produced by cells at the apical ectodermal ridge, which is this little group of cells at the very end or the distal portion of the developing limb bud. Okay, and they signal what is going to be the tip or the distal end of the limb and what is going to be the proximal end, the end near the body, and that is done based on a gradient set up by the diffusion of this growth factor. It's very high at the tip, and that signals develop of digits. In the middle, it's this is a simplification, but is a good overall model for how it works. In the middle, if we have a middle level, we get the formation of wrist bones, and then the two, the radius and the ulna, and then the humerus, right? So those different signal for the development of different bones at different levels. And so if we remove the apical ectodermal ridge, we don't get that signal, and we only get the most proximal parts of the limb developing, and even they don't look quite like they should. So removing that signal negates the proper signals for growth of the limb. If we graft an extra one on there, so we have two competing signals, we get an extra, it's in the same orientation, but we get an extra set of distal elements. So basically you get extra wrist bones, maybe even fused an extra radius and ulna, and extra digits. Okay, this was originally done in the chicken, or at least these sets of experiments are diagrammed in the chicken. Okay, so understand the role of the AER. Again, I don't care that you remember the name, but it sends out a signal to pattern the proximal to distal axis of the developing vertebrate limb. And then you should be able to know what happens if we remove it or if we have an extra version. Now, one other good vocabulary word, ectopic. That's a term you're familiar with, and it's actually used in a very similar manner. So you've probably heard of that in terms of an ectopic pregnancy. So that means a developing embryo that is growing in the wrong place. Ectopic means wrong site or wrong place. And obviously that can have some fairly severe consequences for a developing embryo. So if an embryo implants in the fallopian tubes instead of the uterus, it's ectopic, and that is usually very, very dangerous for both the mother and the developing embryo. Okay, but in this case, we're referring to ectopic cells, cells that are in the wrong place, and so therefore giving a duplicate signal. Okay, so the next one that you need to know that was discovered a little bit later, they called the zone of polarizing activity. Okay, so here's our AER, right? That is critical for correct proximal to distal development. But then they found another set of cells that was found in the posterior region. So here it is. It's kind of internal there, the group of cells there that they've diagrammed. And they found that when they did an ectopic transplantation of this, so they take it off of one chick and put it on another chick that's not damaged, that they ended up with mirror image ectopic digits. And so just like the AER signals proximal to distal, this zone of polarizing activity signals what side is going to be the proximal side, posterior side, the tail end basically of the limb, or the hind part of the limb, and then the front end near the head, the anterior. So zone of proximal activity is responsible for keep doing that, let me be clear. The zone of polarizing activity is responsible for posterior to anterior patterning of the limb, where the AER is responsible for proximal and distal patterning of the limb, getting those gradients set up correctly to create the right axes of the developing limb. And so when we mutate it, we get mutations. And so if we remove this, we get kind of this malformed limb. It doesn't picture it here, but this malformed limb that doesn't have clear anterior or posterior sides. Now, again, there is a very specific morphogen that has been identified, the main one, and then there are cascades of other ones that are involved also. Sometimes it's more complex, but the morphogen here, again, I'm not going to really hold you for this. Don't worry about memorizing it. But the morphogen is called, this is a fun one, sonic hedgehog. Originally discovered in a fruit fly. One of its mutant phenotypes in the fruit fly is the embryo has all these really large, stiff bristles that make it look a little bit like a hedgehog, and it was a more pronounced than another one that looked like a hedgehog. So I don't know if he was a video game fan, but they called it Sonic Hedgehog. Again, many of the fruit fly names are very creative. Okay, so be familiar, review AER, review the zone of polarizing activity. AER, by the way, is apical ectodermal ridge. I usually just say AER, but you should recognize both the full name, AER, and the abbreviation, okay, and ZPA, zone of polarizing activity. Okay, so that is an example of development of the vertebrate limb and the morphogens that are responsible for those early patterns. Okay, now, remember the three types of genes, quick review, right? Housekeeping genes are generally involved in all cells at one level or another at different times maybe, but they're kind of necessary for proper cellular function for all cells. We then, of course, have the specialized function or tissue-specific genes that are expressed only in some cells and usually are not important for development. And then finally, we have these toolkit genes. And so we're beginning to describe and elucidate and go into more detail about these toolkit genes. Now, as we've done this and as we begin to describe all the toolkit genes, here are some of the things that we have found, general trends among the toolkit genes. Number one, it's actually a relatively small percentage of genes. Usually just a few hundred out of, humans have somewhere in the neighborhood of 26,000 genes that have been described. There may be a few more, but a small percentage of those genes. So maybe as few as 1% of all genes are developmentally important genes. And as we already mentioned, they are transcription factors or signaling pathways that have an impact on gene expression in cells. And that their spatial and temporal expression, so location and timing of their expression, is highly correlated with the regions that they are causing to develop. And if we change those, we're going to have drastic impact on those regions, right? They're often named by the phenotype. We already talked about that. And then this remarkable quote-unquote surprise is that many of these are incredibly conserved. They're conserved over hundreds and hundreds of millions of years of evolution and still have the same basic function.